Hello, everyone. Um if you're here and if you're watching me or listening me, so hello. And this is a live session for bioinformatics, for bioinformatics research, for bioinformatics tools, how to use, how to work with, or if you have any other questions related to your research, your current technology, and whatever, you can come and discuss. It's almost 4 p.m. on GMT plus 5. So I'll stay here for some time. And if I'm joined by some of you, then it's okay. We will proceed. Otherwise, I have to disconnect them. Hello again. If you are live with me, then if you want to ask something or if you want to discuss something, you are welcome. So I'll be here for some time. Hassan Ahmad Khan, he's asking me that no one is here. So I have right now two guys available, and I'll be here for 10 more minutes. Okay, let's do this way. Who is the second one? One is Hassan, and who is the second one I have? If you have any question, please ask, or at least you can introduce yourself. Research consultant. Assalamu alaikum, sir. Wa alaikum salam. Research consultant, if you have any question or if you want me to discuss something, you can, because this live session will go live, um, or it will be on the channel, so you and other can watch later also. So far I am here since last 7 minutes or 8 minutes. Then I have to wait for 2 more minutes. Then I have to quit it. Zahid Saeed has asked about molecular dodging. I think you mean molecular docking. A year ago question, and Zahid Said, ex-student. Yeah, I know you are my ex-student, and your question is about molecular docking. So right now on the channel, I have covered almost so many videos, and among them, docking is one of the area I covered extensively, and still I'm covering extensively. I taught on this channel how to perform docking in offline setup, in online setup with Google Colab, and since last 1 month, I'm teaching guys, or you guys, like how to perform docking a protein and ligand using AI or using AI tools this way. So I think we have discussed so many things, are so different, so many different aspects of docking. So exactly, if you are interested in some specifically um, some area of docking or how to do that, like that kind of question, then I can talk on it. But apparently, if you are asking what is molecular docking, so it's just the calculations or the checking or the analysis of different compounds against certain protein for the prediction of their binding affinities. This is called molecular docking. Okay, I think that this time is very odd, and because I did not see anyone here, so let's let's stop this session or make it formally end. Mm, Thank you so much, Mr. Zahid Said and Mr. Hassan, for being with me. And we have to stop this session now because it doesn't make any sense. You're watching me, and I'm watching. I'm waiting for your replies. Okay, Shiv. Hello, sir. Hello, Shiv. How are you? So I was about to quit or to stop. So if you have um, any question, you can ask. Yes, I want to run peptide library for protein docking. Yes, Shiv, you want to run a peptide library for protein docking? How I run, which software I can use? Shiv, I have covered technically, if I tell you on this channel, I have covered three methods: AlphaFold, in fact, AlphaFold 3, Bolts, Chi1. So you can use these three methods or any of them you want. So if you go to the channel, you will find these three methods. Okay. Apart from this, I have covered a protein docking using Google Colab. Okay, I have shared the video, the tutorial, each and everything. So you can also follow that one. And if I'm not wrong, the Colab notebook is using Megadoc or Galaxy Doc. I think it's Megadoc. So now go to this channel. I also named those programs if you are interested in the AI based program like AlphaFold, Bold, Sky1, then choose those methods. But if your question is how to use it, then these tutorials are already there, 20 or 22 minutes video for each tutorial. You will learn it very quickly. That's not very hard and fast rocket science. But if you want to do it by a conventional way, not using AI stuff, then I would suggest to follow that protein docking tutorial. It's present on the channel. And tell me from where I download peptide library. I'm looking for protein peptide docking. So that's very strange question you're asking me, that from where I download a peptide library. Because docking is not a blind thing. Like if I tell you take this peptide and try, Then if you are asking me that, okay, I have protein B, and I tell you again, okay, take this peptide and dock. No, you have to select. This is science. You have to select something with some rational design. Why you are docking some certain peptides against a certain proteins? There must be some rationale in your experiment. So you cannot pick any peptide, or if you are interested just to pick any peptide, so you can just Google like peptide database kind of this, and you will find so many different repositories and those things. Just take peptide sequences and from there and dock it. That's it. If you are interested in any kind of peptide, uh, but if you are looking for protein peptide docking, then you must know from the literature study what kind of peptide you are using, you want to use, why you want to use some certain peptides, blah blah, this kind of. Okay, actually sir, I want develop protein interaction. If you want to develop a protein interaction, but your initial question was how to do a docking. Okay, okay, you want to develop a protein interaction, like you are interested to find out inhibitor from docking. That's it. Okay, so once again, if you are interested to find some inhibitors from docking, then what you have to do, the first thing is to do a literature search, because what kind of peptide you are interested in. I give you a very simplest example to make you understand. For example, this is your protein. This is the active pocket of the protein. Okay. Now, if this active pocket is hydrophilic, or this is polar, okay. Now, if you're taking a peptide of hydrophobic nature and you want to dock this peptide here, it will be a mismatch. It will be a waste of time. You are getting my point? Because if this active pocket is polar, then it's better that your peptide must have polar amino acids. So in that case, you have to select some peptides or a peptide library, or you have to generate your own peptide library. But your prime focus will be on those peptides which are not hydrophobic, which are a kind of, I would say, hydrophilic, and vice versa. If the protein pocket is hydrophobic, then you will need a peptide populated with leucine, alanine, glycine, cysteine, tryptophan, these kind of amino acid, because these are hydrophobic. But if you are looking on the other side, then your peptide must contain like histidine, aspartic acid, glutamic acid, serine, threonine, those kind of amino acid. Okay, but if your active pocket is amphipatic, amphipatic means the active pocket which contains the contribution of hydrophobic and hydrophilic. You cannot say polar, but polar is another. But amphipatic means which contains both kind of thing, hydrophobic and hydrophilic. So this is amphipatic. Okay, now maybe your active pocket is polar. Polar means that in this active pocket, this site is heavily populated with positive charge, this site is heavily populated with negative charge. then your preferred peptide will be to have some positive charge on one end, some negative charges amino acid on the other end. So you have to develop your library with this strategy. So first of all, you have to study, you have to learn about the protein pocket you are interested in. Oh, mean if pocket is hydrophobic, then I need hydrophilic nature of peptide. No, 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 no. If the peptide, okay, if the pocket is hydrophobic, let's make it simple. If the pocket is hydrophobic, then you will need primarily hydrophobic peptides or hydrophobically dominant peptides. If protein pocket is hydrophilic, then you will need primarily hydrophilic dominant peptides. The other way around, if your pocket is empiphatic, then you will need a kind of empiphatic peptides. If your pocket is kind of polar, having positive and negative both charges on the different poles, for example, this is a pocket, but this area is positive hardly, and this area is majorly negative. So it creates a kind of two poles. So we call it polar. Okay, so in this case, the most suitable peptides will be kind of polar peptides, which contains some positive charge on one side, some negative charges on the other side. So the first thing is you need to understand the properties of the active site. It will help you to gather, to generate a library, to combine a library, to find compounds from a library, from different databases, and make your own library. That will be very interesting. And you will always be satisfied if a teacher asks you, or if the examiner asks you how you selected these peptides, you will answer, I did not select randomly. I select by this mechanism. I first study the active pocket, then I came and I selected such kind of peptides based on those properties. And then I found that among this my library containing peptides, this one and this one were the best, blah blah. So this is now a logical and rational research design. Don't go for a just take peptides from somewhere and protein from another place and mix it and say this one is the best. No, no, this is the wrong way. I hope I have answered your question in a sufficient detail. And because we don't have so many people right now, so that's why I took time. You're welcome, Chef. I took time to answer you in a lot of with so many explanation. So I think there are no more questions, so I will wait just for a minute or half. Then if we have no more questions, then I will disconnect. Normally, I come online just later in the night, but most of my listeners are from the USA and from the India. So I thought today that why not this come live this time. It will be just probably 5:30 or 5:18 in India, or maybe 5:48, like this. At least they will not be sleeping, the Indians, and also the Americans will be still alive, or people those who are watching my channel from America. So that's why I choose this time. But I think it was a bad decision from me, because from India only chef connected, and no one came from the USA. So next time I will try to come online again, but later in the night. Okay, thank you so much, and you guys for being with me. And I tried my best to answer your questions, and I hope you will enjoy if you are listening later, or if you're listening this live session, or if you're watching this live session later on. So please subscribe this channel, and I need your support. I'm already trying my best to give you the latest and state of the art tutorials. So still, if you have any question, please drop in the comment section. Thank you so much. Bye bye.